Hello, this is Alice Formiga of eOrganic, and I'd like to welcome you to the webinar on biological control of whiteflies in organic squash. This is the third webinar in a series of 4 about biocontrol in organic vegetable production. Today, I'm very pleased to welcome our 2 presenters, Dr. Oscar Lilbert and Dr. Lorena Lopez of the University of Florida. Dr. Lilbert is a professor in the entomology and nematology department, and his expertise is in agricultural pest management, specifically working with organic vegetable growers, primarily on cucurbits, cole crops, and solanaceous crops. He also works on pest control in small fruits. Dr. Lorena Lopez is a postdoctoral research associate, and her research includes regulation of pests that attack organic zucchini squash by using sustainable practices such as cover crops and conservation of natural enemies. She is also evaluating the use of bacterial strains for control of root knot nematodes. So with that, I'm going to turn over the screen control to our first presenter, Dr. Oscar Lilbert.
Good afternoon. First of all, I would like to thank Alice and eOrganics for organizing this seminar series and biocontrol of agricultural pests. Again, today's topic is we're talking about biological control of whiteflies, and I know from probably the last seminar series, if you saw it, you probably think that there isn't much potential for biological control. But today, I think we have a different story to tell. We will be talking to you about the potential for using biological control in organic agriculture. So in terms of outline, I would be giving a brief introduction about whitefly biology and also talk a little bit about its economic importance. We will be discussing some of the common predators and parasitoids that are used for whitefly management. And then I'll talk to you just briefly about some strategies for biological control, common strategies for biological control. And then I'll talk to you about some preference studies with one of the predator that we use, which is Delphastus catalinae in buckwheat and squash. And then I will be talking about Amblyseius swirskii, and then my co-presenter will come in and talk about some experiments that she did with Amblyseius swirskii. So to introduce you to the system that we used, we have used in terms of whitefly management. Before I actually go there, I just wanted to give you a little bit of background on the economic importance. Whiteflies are probably the most important pest that we have in vegetables, especially in squash and tomatoes. And what they do, they tend to suck the cell sap from these vegetable crops, and they tend to transmit a lot of viruses. And also in squash, they cause something which is called squash silverleaf disorder. You can see symptoms of squash silverleaf disorder here. This is a virus that is spread by whiteflies, the cucurbit leaf crumble virus. And in conventional systems, they tend to use neonicotinoids, soil-acting pesticides to manage whiteflies. Unfortunately, we can't use this in organic system, so we have to develop strategies that could avoid the use of pesticide or limit the use of pesticide in organic systems. So again, the system we use here, you can see the predatory mite, Amblyseius swirskii, and this is the whitefly here, Bemisia tabaci, called the meme. And we have the whitefly predator here, which is Delphastus catalinae. The thing about it is that Amblyseius swirskii manages both whiteflies and thrips in the cucurbit system. I'm talking about squash here, so it's definitely a good predator to explore and to try to adopt or to use in your system, since it managed 2 key pests in the southeast: whiteflies and thrips. Thrips can become a problem sometimes in the spring. Delphastus also can be used to manage whiteflies. A lot of the success about Delphastus catalinae has really been in the greenhouse, but we have had some relatively good experience using this externally as well too. Other insects that we have in the squash system is aphids. So you can see the melon aphids are very common here, and we tend to use parasitoids belonging to the Aphidus and also the Lysophilibus species to regulate the population of melon aphids. So other common parasitoids that are used to control whiteflies, Encarsia formosa, and this is used very extensively in Europe, especially in places like Spain and Holland, and to some extent in England. We don't use it a lot here in the United States because it performs very well inside the greenhouse, but outside the greenhouse, the performance tends to fall off a little bit. Eretmocerus is also another whitefly parasitoid, and it's used in the West Coast along with other strategies to manage whitefly populations, both in ornamentals and in vegetable crops. So this one is used a little bit more in the US as opposed to Encarsia formosa, which is used extensively in the greenhouse. But they're parasitoids that can be used to regulate whitefly population. Now, to introduce biological control, I just wanted to do this very briefly so that you can understand exactly where we are and how we use biological control. And there are several strategies for implement biological control into the farm. First, we have the introduction. And basically, we tend not to use introduction very often in organic agriculture because this involves taking an insect, whether it's a predator or parasitoids, from one area and introducing it to a new area. A lot of times we have to actually go outside the country to get these natural enemies and bring them back into the country. And when we do that, one of the problems is that it has to go through an extensive series of testing before we can release it into the environment. So when we think of biological control, we're thinking about using one organism to control another organism. And the strategies we have, again, is introduction. Augmentation involves building up the natural enemy or the biological control agent in the environment. And augmentation can be actually divided into two broad areas we call inundative releases. And inundative releases is what a lot of us tend to do, where we release large quantities of biological control agent, and we don't expect them to reproduce in the system, but we expect them to suppress the pest population. For example, those of you who grow things like strawberries, we tend to use a predatory mite called Phytocetus piscimulis, and that is used to almost like a pesticide to push the mite population down. So again, inundative releases meaning you release large quantities of these natural enemies, and you don't expect any reproduction in the system, but you expect the population to go down. Whereas inoculative releases, you tend to release less natural enemies, but you expect them to reproduce within the system and build up their population and suppress the pests eventually. Conservation biological control is really important, especially in organic agriculture, because what we are doing, we're basically changing the habitat where the biocontrol agent can grow and develop. For example, we may use, and you're going to see quite a bit about this today, some kind of companion plants in the system. And these companion plants tend to provide resources. For example, they provide nutrient resources. They provide places where these biocontrol agents can lay their eggs, and they provide other essential nutrients that the biocontrol agent needs to build up. So habitat manipulation is very important when it comes to conservation biology. We can also do conservation biological control by limiting the use of pesticides. If you limit the use of pesticide in the system, and there are pesticides that are labeled for organic use, so obviously you can use those pesticides. The problem is if you use too much of those pesticides and those that have negative effects on the biocontrol agent, it will reduce the population as well. So what we do, the reason why we can implement biocontrol agents in the organic system is because we use a combination of strategies. For example, we use the biocontrol agent. And for example, if we're using Delphastus, we can have Delphastus there, but not only Delphastus, we may use some kind of flowering plants. And here you can see a field, one of our experimental field where we have marigolds in the center. And the only reason why we are having the marigold there is to attract the beneficial insects or the natural enemies that tends to regulate the population of the pest. And then sometimes when the population is really high, we use what we call an OMRI-approved insecticide or an organic insecticide to knock the population down first. And then after a few days, we release the biocontrol agent, and then we allow the biocontrol agent to flourish within the flowering plants, hoping that the flowering plants are providing the resources that the biocontrol agent needs. So we really integrate the tactics in order for us to allow the biocontrol agents to survive in the system. Here I'm showing you some experiment that we did here, again using the marigold, as I mentioned before, and we also had cowpea. These are cropping systems or intercropping systems that were intercrop with squash. So we have squash, as you saw in the previous slide, and then we have marigold in the center. We have squash again at the ends and marigold in the center. And then there are times when we have a mixture of marigold and cowpea, as you can see here. And MP is the pesticide, one of the pesticides, OMRI-approved pesticide that can be used in an organic system. And then we have untreated plots where we just have squash. And if you were to look at the results we got here, you'd realize that the cowpea here, and again, when you look at these numbers, you'd realize that if you have the same letter, it is not significantly different. The letters have to be different. So here you can see that when you compare marigold, cowpea with the control, cowpea had significantly more parasitoids than the control. And the parasitoids we are talking about, these specific parasitoids were Encasia formosa and Aphidus. This is used to control aphids in the system. They were, and if you were looking, I guess this was done in 2016, this was in 2017. In 2017, again, you can see marigold here had more parasitoids, again, than the control. So you can see intercropping in sometimes do harbor these parasitoids and increase the population. And this benefits us because it reduces the pest pressure in many times. So first of all, I'm going to talk about some experiments with this whitefly predator, which is ladybug, Delphastus catilina, which I was talking about. And one of the reasons why it is considered as maybe a good biocontrol agent for whiteflies management is because it's a specialist on whiteflies and feeds strictly on whiteflies. It has a high consumption rate. In other words, it consumes quite a bit of whiteflies. I think there are thousands of whiteflies can be consumed in one season. It has a long adult survival. The adults tend to live 60 days or higher. And reproduction rate is really good, and it's also commercially available. So this is a good potential biocontrol agent for us to use for organic agriculture. Here, I'm talking about some experiments, and this was done in the greenhouse and then in the field, where here you can see we have buckwheat, and in the greenhouse, we have exclusion cage with squash, and buckwheat and also whiteflies are in the cages. And then where the experiment is in the main plot, we have in some plots we have the predator, which is Delphastus catilinae, and in some of the treatments we did not include the predator. In the subplots we had squash in some plots and buckwheat in some plots. and so forth. Again, experimental design had 4 replicates. And basically what we found here, again, the results here shows the months when the experiments were done and the population of whiteflies, adult whiteflies on the y-axis here. So the treatments were buckwheat with Delphastus catiline, squash with Delphastus catiline, buckwheat without the catiline, and squash without the catiline. But what we find was that from this experiment that we did not find hardly any whiteflies in the buckwheat. And because we hardly find any whiteflies in the buckwheat, obviously Delphastus did not stick around too long. So what you're seeing on the graph is primarily the results from the squash. But you can see here squash without the Delphastus catiline, where the population tends to rise. I'm talking about the adults whiteflies here. The population tends to be very high. When we introduce the predatory or the predator, what happens? The population drops significantly. And again, for the adult whitefly. And we look at the immature whiteflies, the results were even more dramatic. You find that without the Delphastus catiline, the predator, population of whiteflies was really high, reaching almost up to 300 immature whiteflies per plant. And when we introduced the Delphastus catiline, the population dropped significantly all the way down to about 75. So you can see that the Delphastus catiline had some effect in terms of dropping the population significantly here. One of the things that we do when we have very high populations, I think I mentioned this earlier, of whiteflies, we tend to reduce the population or reduce the pressure by using organic pesticides. Pesticides are labeled for organic use. And here we screened quite a few pesticides, and you can see Pyganic, MP, Isosol, Entrust. And we put Entrust in there as a standard because a lot of the growers, they tend to use Entrust for almost everything. I mean, Entrust is a pesticide that is used almost for every single thing with organic agriculture. So we wanted to use that as a standard. MP and Pyganic, we wanted to look at those. But basically from the results, what we found, the best performing pesticide, as you can see, was Pyganic. And then this was followed by MP. But when we did some additional experiments, we realized the Pyganic lasted only for a few days. The effectiveness of the Pyganic only lasted for a few days. So I think a lot of our future recommendation went with MP. So the question is, when can you release the predator in the field, Delphastus catiline, after you use the pesticide? And here we have the Pyganic, we have the MP, and we have untreated. And you can see 1 day after release, 3 days after release, and 5 days after we spray the Pyganic. And again, you can see MP here, 1 day release after spraying MP, 3 days after spraying MPs, and 5 days. And basically what we have found, this is untreated. In other words, it's controlled for the experiment. And what we want to see high numbers of Delphastus catiline. And you can see that when we release the predator 5 days after, this was not different at all for the control. Like 3 days wasn't different also. There was a slight numeric reduction, but nothing significant. So 3 days, there was some reduction. 1 day was definitely significant. So if you spray today and you release the biocontrol tomorrow, you would get some reduction in terms of your biocontrol agent. And we found almost the same exact results for MP. Again, if you spray today and release tomorrow, 1 day after, there's a significant reduction in terms of Delphastus catiline. 5 days after, there's hardly any difference. And 3 days after, there was some reduction, but not significantly different to 5 days. I'm going to switch now and talk about Amblyseius swirskii. And again, this was a predatory mite that we have been looking at very seriously because it has done some good work for us on thrips in the squash system, and we wanted to look at its potential for whiteflies as well. And a lot of my co-presenter information from Lorena Lopez will talk about Amblyseius swirskii as well too. But it attracted substantial interest again because of its efficacy on thrips, and from the experiments that we did, we realized also that Amblyseius swirskii tend to prefer eggs and first nymphal stage of whiteflies. There are many different formulations that are available commercially when it comes to Amblyseius swirskii, and these are two different formulations. Here we have, as you can see here, and the experiment will explain all this that I'm about to talk about. So we have three different cultivars of squash here. We have Zephyr, 8 balls, and Sunburst. Now this is directly related to the experiment that we were supposed to do for the grant. And some of these results you saw last week with the presentation, Babu from Georgia, professor from Georgia, who talk about their results. Our results are a little bit different to their results. So here we're trying to figure out here, where is the best place to release these predatory mites? Again, so we release some on the leaf, we release some on the stem, and then we use these chassis, so to speak, which is available commercially. And then we have untreated squash here, squash which wasn't treated with any predatory mites. And again, we have 3 different cultivars. So when we sampled, 2 weeks after, and this sample was done on a weekly basis, but 2 weeks after, we were still catching more Amblyseius swirskii, or the predatory mite, on the leaves compared to the sachets and the stem and the control. We think the reason for this is because, when we sample, we were sampling mostly the leaves, and this may be the reason why we had higher percentage in our sampling counts on the leaf. But the interesting thing is that when we look at the whitefly population, is that whether the releases were done on the leaves, the sachets, or the stem, it reduced whitefly population to some extent compared to the control. So we did get some reduction in terms of whitefly pressure when we release, again, whether it's the leaves, sachets, or the stem. There were no significant differences between all 3. When we look at virus infected, and I know you're still seeing some pressure here. For example, on the leaves, we counted, this is the susceptibility of organic squash to cucurbit leaf crumble virus. And you're seeing here that in the control, we have significantly higher number of plants infected with the virus, almost 5.5 compared to 2.5 here plants. And then again, whether the releases were done on the leaves, the sachets, or the stem, it did not make a difference. We saw a reduction in terms of the number of virus infected plants. And not only that, the important thing is that there were no significant differences between any of the treatments, whether they were released on the leaf, sachets, or the stem. In terms of the varieties that we look at, whether this is significant or not, I'm not too sure yet. Sunburst had the highest population of whiteflies. Maybe this cultivar or this variety was just more attractive than the others. But this is really important, the marketable yield here. You'll find that, and this is what most people are concerned about, whether the releases were done on the leaf, the sachets, or the stem, we had higher yields in terms of the kilograms of marketable yield for squash compared to the control. So again, you're seeing some reduction here using the predatory mites, and you'll probably see a lot more on this from my other presenter. So some of the summary and findings that from these studies was that Delphastus catiline, which is the predator, reduced whitefly population in organic squash. The pesticide, or the organic pesticide, MP and Pyganic, were the best performing organic pesticides, obviously from our screening, because we looked at all 5 pesticides. And they were also compatible with D-catiline, which is the predator. And as I indicated earlier, that these insecticides can be used 3 or 5 days after spraying the pesticide, so to speak. The work that we have done have shown that the use of companion plants increase the number of natural enemies and also conserve them to a great extent. Where we intercrop things like marigold or other types of companion plants, we had higher numbers of natural enemies there. Amblyseius swirskii reduced whitefly population below the control. That was quite obvious in the experiments. And irrespective of the release method we use, whether it's on the stem or the leaf, we saw pretty much the same results in terms of reducing viral infection, increasing yield, and so forth, and reducing whitefly pressure as well. I'm going to turn my presentation over to Lorena Lopez, and she's going to be talking about some of the experiments that she did looking at Amblyseius swirskii.
So hi, everyone. So I'm going to talk a little bit more in detail about one study where we evaluated the effect of intercropping flowering plants together with the releases of the predatory mite Amblyseius swirskii. So basically what we wanted to do here was to combine two tactics for pest management. So we wanted to intercrop zucchini squash with African marigolds and sweet alyssum, and we wanted to know which of these two companion plants will perform better as companion plants. And together with that, we wanted to see the performance of Amblyseius swirskii released on the squash only for them to feed on the whiteflies. The companion plants were there to attract natural enemies and complement that biological control service, but also to help suppressing other pests like aphids, because the predatory mites don't feed on the aphids. So we set up, okay, there you go. So we set up small field trial in a research center. Wait, this is just going crazy. Apologize for this, but it's not. There you go. So we set up a field trial in a research center, and the plots consisted on 18-feet plots with 3 raised beds. The outer beds were planted with squash only. And then the treatments were defined based on what was planted in the middle, what companion plant was planted in the middle bed. So, and also this was evaluated in the absence or presence of the predatory mite Amblyseius swirskii. So, for example, in treatment 1 or 2, the treatments were based on either marigold or sweet alyssum planted in the middle bed with no predatory mite release. Treatment 3 and 4 was either marigold or sweet alyssum planted in the middle bed with the release of the predatory mite only in the squash in the outer beds. Then we have a treatment 5 with no companion plants, but the release of swirskii on the squash. And then we have 2 controls. We had one with no companion plants, no predatory mites, but the spray of NPED on the squash. And then one last treatment with no pest management at all. And we set up yellow sticky traps over a 5-week period. And we recorded the number of adult whiteflies per trap, which is here. Over this 5-week period, we started 3 weeks after planting the squash in the field. We also recorded the number of predatory mites, here are the treatments. And even though we have commonly higher pressure during the fall here in North Central Florida, this was spring season, because that year we had higher pressure on the spring instead of the fall. So that's why you see this pattern of whiteflies going up as the time goes by. And what we saw was that when we, after releasing the predatory mites on week 4, the number of adult whiteflies was significantly lower in the treatments where the predatory mites were released alone with no companion plants in the blue bars, and also where the predatory mites were released together with intercropping sweetylism in this yellow bar, and that was true for these first 2 weeks, and a little bit here and in the last week of the season. So they reduced the number of adult whiteflies. I also checked the squash silverleaf disorder that Oscar mentioned. This is caused by the feeding of whitefly immatures in the squash, and when they cause the leaves to go silvered, to start going silver. And I rated the plants using our arbitrary index from 0 to 4, 0 being no silvering at all, and 4, all leaves in the plant were silvered. And there was significantly lower squash silverleaf in the plants in the treatments that were sprayed with MPED. That was the only one lower, but in the case, for example, the marigolds only, it was really high silverleaf disorder, and it was comparable to no pest management treatment, which is not good. So when we go to the yield, we had significant differences, but only the treatment where MPED was sprayed on the squash was significantly higher compared with the other treatments. So there was a little bit of noise in the treatment. So the marigolds, it was hard to see if the marigolds in combinations with the predatory mites had a significant effect on the yields here, but they definitely had an effect on pest populations, whitefly populations. So we wanted to compare which of these two, based on these studies, which of these two companion plants perform better. And what we learned was that sweetylism and also marigolds were not used by the predatory mites. The predatory mites did not establish on those companion plants. They didn't move towards them. In the case of sweetylism, they were not infested with aphids or whiteflies. And actually in the squash intercrop close to the sweetylism, there were very few aphids. Whereas for the marigolds hosted a lot of aphids, and there was a spillover effect on the squash because the squash around those marigolds had more aphids. In the case of sweetylism, they were attracted to predatory flies and minor parrot bugs, that is, as it was suspected. And in the case of the marigolds, they were attracted to, especially to minor parrot bugs. And it seemed like they were attracted to the thrips because the marigold flowers were loaded with thrips, whereas the sweetylism hosted very few thrips in their flowers. So based on this, it seems like sweetylism was performed better as a companion plant for squash because it didn't host any pests. It was still attracted to natural enemies. And whereas marigolds, they seem to perform better as a trap crop. Sometimes it's needed, like for aphids, if your major problem is aphids or thrips, but further research needs to be done to clarify that. So we decided to see what would happen if we put intercrop, like bigger strips of sweetylism, again, together with a predatory mite in a bigger field, in a large scale scenario. So we set up an on-farm trial with a bigger experimental area where we wanted to evaluate the effect of these two techniques, the predatory mite release and the flowering strips of sweetylism, and see their impact on pest and predator distribution in the experimental area, their effect on pest suppression and squash yield as well. This is not moving. Okay, there you go. Besides having flowering plants there, we also wanted to know if the predatory mites were able to move around in different squash cultivars. Do they have any preference? Do they have any limitations? So we used 3 different squash cultivars for this trial. And I want to mention that this was an organic farm where we tried this. Bear with me, it's a little slow. So this is the experimental setup. So we had two crops, two plots, one control. Each of these color lines represent a different squash cultivar. There was no sweetalism in the control, no companion plants. And then the treatment had 25% of their area planted with sweetalism in these yellow patches. And then in the middle, we released the predatory mites in the squash. We wanted to see their movement. One thing that is important to mention here is that we didn't apply any pesticides to control the whiteflies or the aphids. We only applied one cover spray of Anthrus to both plots once in the season to control melonworms. That was it. And then from these plots, I took a bunch of sampling points from this area and measured the number of whiteflies and predatory mites. And then I was able to follow the distribution pattern of the predatory mites and in the bottom, they're mapped in the bottom. And the prey, remember that whitefly eggs are preferred by these predatory mites, and I mapped it here in the top. This is the control plot, the map for the control plot, and the map for the treatment. And I did this for a 5-week period. I'm not going to go into detail for this. I just want to show you how we follow their distribution. So the blue, the darker the blue, the less whiteflies or predatory mites we had. And the yellow or closer to red, the more whiteflies or predatory mites we have. So I released, the first week I used sachets because as Oscar mentioned, there were no differences between that. But I had an issue with the sachets, not because of the formulation, but the firing ants started getting into the sachets and eating the mites and the bran that comes with it. So we had to release the second time, the second week. You can see here in the center, you see high numbers of predatory mites because that's where we released them. And this time we used bran on top of the leaves and that worked fine. And you can see very rapidly that they dispersed within 24 hours. I released and the next day I sampled and they were all over the place. And they even start moving towards the control because there were so many whitefly eggs in this area. And then in the third week, you can start seeing how the number of whitefly eggs start to go down where the predatory mites were released. And this is true also in the fourth week. And you can see that they start moving around very easily among the cultivars, squash cultivars. And then the last week, they're very low, both of them, probably because it starts to get chilly here. This is very important because we were able to see the effect of this predatory mite over time on whitefly eggs, on whitefly populations. And over all this time, what we also found is that whitefly eggs and immatures were significantly lower in the treatment where the predatory mites were released. And also, the silver leaf disorder caused by immatures was significantly lower in the treatment where the predatory mites were present. And this is pretty much, the treatment was close to index 2, which is like very few silvering, whereas the control was closer to index 3, that where you can see more silvering of the leaves. We also counted aphids because the main purpose of sweetalism there was to attract predatory flies and predatory other predators that would feed on the aphids, since the predatory mites don't feed on them. And it seems like there was an effect of having these companion plants there on aphid population because they were significantly lower where the sweetalism was. We found a mix of different aphid species, but most of them were melon aphids. And then finally, for the yield, we had no significant differences among the treatments, unfortunately, but there are two things that are important to remember here. And one is that we had 25% of the area in the control treatment planted with squash that we didn't have in treatment because we had the sweetalism. So that may have contributed to the non-significant result here. And also, even though we didn't have higher yield, with the numbers of aphids and whiteflies that we had in the treatment, it eliminated the need to apply any pesticides. So that will be translated into lower pest management costs. So to summarize, the predatory mites Amblyseius swirskii established and dispersed successfully regardless of the squash variety. And this is really important because this is one of the first of the very few studies evaluating the performance of this predatory mite in squash and in the open field. Most of the time they're used in other vegetable crops like peppers or under protector structures. But we were really happy that they're established and that they disperse very easily in this squash field under these circumstances. The sweetolicin combined with the predatory mite had a significant reduction effect on whitefly populations and aphid populations, and also the silver leaf disorder. Also, these combined practices reduce the need to pesticides. Even though we didn't have differences in yield, this can be translated into reduced pest management costs. And this is pretty much the whole point of the study, is to reduce costs and also to combine this biological control in cultural practices in organic squash. So with this, I'm going to finish. And I want to thank all the members of the Small Fruit and Vegetable IPM lab that helped conducting all these studies that we just talked about. I want to thank eOrganic and Alice for helping us put in this webinar together. And of course, all these studies were possible thanks to the funding of the ORAI grant by USDA NIFA. And thank you all for attending our webinar.
Okay, yeah, so we had a question during the webinar about how you apply the Amblyseius on cucurbit stems without the sachet.
From what I see, they were applied at the base of the stem. So instead of on the actual stem itself, they were at the base of the stem, but it did not matter. What we found out was that as long as the Amblyseius swirskii was applied, they can regulate the pest population. I don't know if Lorena, who actually did some experiments with this, want to say anything about that, but they were applied at the base of the stem, those that I've seen. Yes. Lorena, you want to say anything about it? Yeah.
I can add a little bit more details if you need more how to do it. So the way we did it was when you buy the bottle with brand, it pretty much says that it in 1 milliliter of the brand, you can find around 50 to 100, depending on how big the bottle is, 50 or 100 mites. So basically you want to calculate first, check the brand a little bit, sample of how active the mites are, just to make sure that you have a good amount of mites, because sometimes they don't come very healthy. Just check that, and based on those numbers, you can calculate the amount of brand you need on each of the plants. If you want a rate for high infestation, if you want a rate for medium infestation, you can check the rates online from the company that you buy them, and then with those prior checks under the stereoscope, you can make sure how much do you need for each of the plants or each of the area that you need. That's pretty much how we did it.
Okay, thank you. Let's see. We have another question about how you know whether the whitefly, aphids, or thrips in the experiments weren't affected also by the presence of nitrogen fertilizer in the soil.
Because we have a control that was treated the same way like all of the other plants. And the only difference between the control and the other plots was the treatment that we have in the actual plot itself. So we are actually seeing treatment effects as opposed to the effects of the nitrogen, because all of the plants receive the same amount of nitrogen. Yeah. Okay. When you look at the results between the control and the treatment, you can see the difference. Yeah.
Okay. Yeah, that makes sense. Thank you. Okay, and have these results been published yet, or are you planning to publish them?
Very good question. Some of the results, those that I presented, has been published, but Lorena's work that she did for a PhD has not been published yet. So the work that Lorena talked about has not been published yet, and that's one of the reasons why we have some reservation in the amount of information we share. But the first part of all of my presentation has been published. Yeah.
Okay, that's great. And maybe what we can do is put a link to that publication on the webinar
Yes.
page there so that people can see that and easily find it. So if you look on the eOrganic webinar archive, if you just Google webinars by eOrganic, we'll do this after the presentation, and probably by the end of the day, it will be up there underneath this webinar's information. So we have a question about whether or not there are some predators of squash bugs that you know of.
Good question. No, I was going to say that squash bug is not a problem in Florida. So for that reason, we don't tend to work on squash bug. But I know there are some general predators out there. For example, I know things like big-eyed bug and also the minute parrot bug. They tend to feed on the eggs of those squash bugs. But in terms of one that has involved quite a bit of research, I don't have that information right now because it's not a problem here in Florida, where we work in Florida. Yeah. Did you want to add something, Lorena?
Yes. So as Oscar mentioned, here's not a major issue, but in the north, in Georgia, and in other states up, there are parasitoids that are being used to control that pest. They're egg parasitoids for them. And in terms of predators, I know there are a few predatory steam bugs, like the spine soldier bug that feeds especially on the immatures of them. But what I've seen so far is that it's mostly use egg parasitoids to control them more than predators.
Okay, great. Thank you. Here's a question related to Dr. Lilbert's summary page. It says he stated that insecticides can be applied 3 to 5 days after field deployment of, but during the presentation, the emphasis was on the application of the biopesticide before the introduction of D-Cataline. And so if you could clarify that, that would be great.
No, well, for organic agriculture, pesticides can only be used as a last resort. So we have to explore all of the cultural and biological control information or strategies before we can use pesticides. So in cases where we have very high population of a pest, the whitefly, for example, and we really can't suppress the population using a biological control agent, we have to come in first with a pesticide to knock down the population, and then we can release the biocontrol agent. So basically what I was showing you is that we can use pesticides in some instances to knock down the population, and then you can come in later and release a biocontrol agent, but this has to be done 3 to 5 days after.
Okay, hopefully that makes sense. And if you have any further questions, feel free to type those in as well. Okay, let's see. The next question is, oh yeah, someone asked about links for biological control agents and where you can buy them. I believe, Lorena, you already typed in some links into the chat box here, and I think it went to all, yeah, it did. It went to all the panelists and attendees. So if you open up your chat box and you sort of scroll upward there, Lorena sent some helpful information and guidance on finding and purchasing biological control agents. And if you click on those links, you should be able to find those. So yeah, thank you for doing that. Let's see. Question further. Let's see, I can't. Oh, okay. If I just want to understand the question. If we have to spray, what would be the best treatment to kill pests but not to kill beneficial insects? You did address that a little bit there.
Well, if we are trying to manage whiteflies, the best product we have, based on the research we have done, is MPED. You get comparable results from using Pyoganic. The problem with Pyoganic is that the efficacy only lasts for about less than a week, to 3 or 4 days. With MPED, the efficacy tends to last a little longer. So we recommend the use of MPED for whitefly management. Yeah. A lot of the growers tend to use Entrust, but based on the results we have, in fact, we are advising growers in Florida not to use Entrust for whitefly management because it really doesn't work. Yeah.
Okay, thank you. Yeah, that's useful. It says, okay, the question is, even though you stated the use of an arbitrary index for scoring silverleaf infection, can you share some more details on some general guidelines that you used in scoring the infection?
Yes. Lorena, jump in because I know you just recently did that. Yeah.
Okay, so I just sent a link to the chat to everyone to see. This is a very popular publication to measure silverleaf. That's the scoring system, the arbitrary index that I mentioned is described there. It's kind of old, but it's the one that it's mostly used. And what I recommend is that if you follow that scoring system, is that the same person should do it every time if you're sampling every week, or at least try to, or at least work with the people that you're working to make sure that you all have the same rating perspective, you could call it, because I mean, it can change from people to see it because it's very subjective, but it's the one that it's more popular and the ones that works best when you're actually doing this kind of scoring.
Okay, thank you. Yeah. Okay. We have a question about whether or not you used alyssum transplants or whether you direct seeded them.
Here in Florida, when we grow in the field for experimental purposes, we normally do direct seeding. However, there are growers here that do transplanting as well. I think the grower that Lorena worked with actually did transplants, right, Lorena?
Yes, he transplants all the time. Yeah.
Yeah. So we do both. We do direct seeding and transplants at the same time. Yeah.
I believe Babu last week also mentioned it depends on where you are.
Yeah.
Like here we have the ability to to seed out and it's fine. But he was mentioning that in Georgia they have to transplant because otherwise it will be as fast as they emerge from the seed, they will be covered from whiteflies, and that's an issue.
Yeah.
So
up in Georgia. Yeah.
They had incredible pest pressure that Babu talked about in the last webinar in his area in Georgia. So yeah, that was pretty impressive when he talked about that. Okay, so another question. Assuming one is using organic pesticides that are approved, would you recommend alternating pesticides with biocontrol?
Well, it all depends on the crop. If the crop lasts for a very long time, like if you're growing something like strawberries that's in the field for 6 months, you have time to do alternating the pesticide with the biocontrol because the crop is in the field for 6 months. The problem with squash, it's only in the field for like 8 weeks, 6 to 8 weeks. And if you try that, by the time you're ready to use the biological control, you wouldn't give it sufficient time to get established in the system. So I do advocate the use of alternating pesticides for management of any insect. However, I will not try to alternate a pesticide with a biocontrol in the case of squash because of the short duration of the time the crop is in the field. It's only 6 weeks, 6 to 8 weeks, the crop is there.
Okay, thank you. Here's another question. In Dr. Lilbert's experiment, it looked as though the flowering strips actually increased the pest presence on the squash, but in Dr. Lopez's field trial, it appeared that the flowering strips helped suppress aphids and attract beneficial predators. Could you talk more about that?
In our case, there wasn't a significant reduction in the pest population in my case. However, we had higher incidences of biological control agents, but there wasn't a significant reduction in the pest. We think the reason for this is because the plots were relatively small. I think in a bigger area, we may see more pest suppression. Lorena used a completely different predator. These two predators perform differently. I actually used, if they're talking about my Dalphastus experiments, I think that's what they're referring to. That's a completely different predator to the one Lorena used, and they tend to perform completely different. I hope I answered the question. Yeah.
I would
Lorena,
like
you want
to
add something? Yeah.
Yes, I would like to add something there. I'm not really sure what graph they're referring to, but I know that Oscar mentioned there wasn't a study where they have also cowpeas.
Yeah.
Actually, I didn't show that data, but I also tried cowpeas, and I didn't continue using them because they attract so much aphids.
Yeah.
Like, so it depends on the companion plants. We had to take it out because it just filled all our squash with aphids. And then we moved to marigolds and sweet alyssum. And it turns out that the marigolds also attract aphids and thrips, but they didn't have the same effect as cowpeas. They kept them on their flowers. Like, that's why I mentioned that it could be used maybe as a trap crop. So that's why we were trying different companion plants that could have different effects. And it's like, maybe that's why the cowpeas were there, and what Dr. Leiber mentioned, bigger plots make a difference too.
So thank you so much, Dr. Lovberg and Dr. Lopez, for presenting today and sharing your knowledge with us. And thanks to everyone for joining us.
Thank you. I appreciate it.
Thank you.
Thanks.